I'm Pubudu Handakumbura. I'm a plant biologist at EMSL, and I'm also the team lead for the rhizosphere function team. And today I'm going to tell you an example where we used some of the EMSL capabilities and tried to look at plant microbe interactions at a more resolved way than what's available right now. So just to remind everybody, the rhizosphere, what we define as the rhizosphere is the thin layer of soil that's surrounded by a plant root or directly impacted by a plant root. And the rhizosphere itself is one of the most dynamic interfaces on Earth. It houses numerous inter and intra-kingdom interactions. And we believe all of these beneficial interactions leads to resilient ecosystem. So it provides resilience to plants, the crops, improves soil health, and enables nutrient cycling. So for numerous reasons, we want to understand the rhizosphere plant microbe interactions at a scale that's meaningful for us to use synthetic biology approaches for rhizosphere engineering. However, this hidden half is really hard to visualize and to really take apart. So for our studies, we wanted to understand root microbe interactions at the rhizosphere at a scale that's more meaningful, that is more relevant to what's happening out in the setting. So these are some of the examples where the scientific community has looked into this. This is not something new. There are so many sophisticated tools, platforms out there where we can look at plant microbe interaction. But most of these are at in controlled environments and at much smaller scale. So these are there is a variety of microfluidic platforms and small benchtop scale ecosystems that scientists use to study these interactions. However, now these systems are constrained by how big of a system that you can use, or they are either constrained by the developmental stage that you can study, and sometimes, you know, like types of interactions, plant microbe interactions you can study. However, these do generate amazing insights into single cell level insights. But for our purpose, we wanted to understand, can we look at a soil grown plant? Can we look at those interactions in a more native setting? So for that, we came up with this RhizoGrid root cartography workflow. So this was initially an EMSL investment, a strategic science investment, where we started building these RhizoGrids and trying to see how we can best capture the root system in a native environment. So this starts with the RhizoGrid itself. So like these are 3D printed grids that's fitted in a pot. So you can grow your system in one of these pots. And then we use the XCT capability at EMSL to capture the root system non-destructively. So we will capture our root system, and later on, using those XCT data, we can reconstruct the root system. So you get details about the root system architecture and much more from these XCT-derived reconstructed root images. And once we've captured our roots, then we can go in and harvest our roots sequentially from this system. So the beauty of this, you can customize it depending on your studying, and also how many layers of grids you want to have in the system and the growth media. So in the examples I'm going to show you, I have used a local field soil for our experiments to mimic something that's closer to what's happening out in the real world. So once you have captured your image, we can harvest the roots, and the the soil that's surrounded with around these root pieces, which we call our rhizosphere soil, as well as, like you can harvest your bulk soils from each of these pieces. So once once we've imaged, this is destructive sampling, we can extract metabolites from the roots and the rhizosphere. We can also extract the microbial DNA, and then we can use amplicon sequencing, we can use metabolomics. And in these examples, I've used these techniques to look at and to build these metabolite-microbe networks, and finally to map them to where they are coming from from the pieces of the roots. But with this same setup, now once you have this indexing, you are able to do other beyond metabolomics and amplicon sequencing. So you can also do RNA, root tissue. We have done examples where we have done labeled inoculations of microbes and looked at cell sorting. So you can look at how the fluorescent signals are distributed along the root system in these different regions. We have also used it where we have mimicked a drought situation, which I will show you some preliminary data at the end, to see, like, can we realistically mimic a drought scenario and generate data that's field relevant. So this is a whole workflow. Starts with planting your experiment on RhizoGrids. And just to show you what these things look like, so this is, again, like, it's a 3D printed unit. These are modular units, so you can stack them and customize your setup depending on how deep of a pot you need. And we have marked each depth into 4 quadrants. We call them ABCD. We've labeled them ABCD so that it's easy for us to index each sample based on the depth it's coming from and the quadrant. So within that 3-dimensional space, you are now giving coordinates. So each of these samples you extract from this. And this figure C is a top view or XCT scan where you are able to see your pot, the 3D printed grids, and the soil medium you have enough resolution to distinguish between the grids, the pot, and the soil and the roots. That is important if you are interested in generating and reconstructing the 3D root system from this approach. And this is one of my sorghum plants. And this is a reconstructed image of the grids and the roots. And so after Anil, he's a scientist at EMSL, so he has been working with us in generating these root reconstruction. And Thomas, who's a co-chair this session, leads this capability. So he'll be able to help you with more detail on XCT. So we are now able to visualize our roots in 3 dimensions, and we are able to capture most of the root system. Of course, there are constraints. Even with this resolution constraints, however, we can reconstruct majority of the root system. And so, like I said, we've harvested our root tissue from each of these sections. So this is, I've labeled them one through, however many depths that you have, one always being closer to the top or the crown of the plant. So you can see all these numbers correlate to the positions of the soil depths. And in this principal component analysis, so we are looking at the overall metabolome of these root tissues generated from each of these different depths. And the numbers correspond to the depths, and you start to see that there is separation between the metabolomes as you go from top to bottom of the roots. And in figure C, we are again looking at few of the selected metabolites out of the the thousands of features we generated using LC. Again, this is a heat map where you are looking at the relative abundance of each of the metabolites and one being highly abundant. So you start to see, right, along the route from top to bottom, there is a gradient. And even between all of these metabolites, you start to see there are so many differences. So this is what we want to understand a little bit more closer, because the conventional way of just extracting the whole route averages out all of this information that we can now capture more spatially. So this is work done by Albert. And then Ryan took all of the microbial amplicon sequence data, 16F sequence data from all these samples that we derived. And he also took the metabolite information, where we represent metabolites as gray triangles in this network analysis, and all the microbial information as circles, and they are all colored by the genera. And we started to put these metabolite-microbe co-abundance networks to understand what are the more important nodes in this. Again, like the overall picture is much more complicated than this. These are like some extracted subnetworks. Just to show you, there is so much connection to metabolites, microbes, and this is looking at the overall picture, not even considering the depths. This is just taking the whole metabolome, taking the whole microbiome, 16F data, and asking like what can it find on. And then Winston took all of that data again and did a redundancy analysis. It's more like a constrained principal component analysis. And so in this, metabolites are labeled as red text, microbial genera as black text, and the soil depth in blue, increasing in color. So top again, one is our top layer closer to the crown. And we are trying to figure out, like, are there metabolite microbe connections that are specific to each of the soil depths? And yes, that is something we find. They are clustering based on the soil depths. And so, for example, if you look at here, we find quinate and few microbial genera associated with a specific depth. So with this was a replicated experiment, and we are able to find there is so much heterogeneity between our biological replicates. However, we are able to find common threads between these biological reps. And the RhizoGrid indexing and sampling is specific enough that we are able to connect metabolite microbe interactions to specific locations within this depth. And then we also, one aspect of this work is to really visualize all of this information along the routes. So that's what Kate did. So she took all of this information, she wrote a Python script. And so this is the reconstructed game that we saw before. And she took all the metabolite information, the relative abundance, and she was able to map them along the route. So in this specific example, we are looking at the relative abundance of one metabolite, aconitic acid. And you see it's highly abundant closer to the lower depths. And then we also looked at one of those microbial genera. And again, a larger circle represents higher abundance. And you see they are more abundant closer to the lower depths, again, of the growing root. But again, these dots are just showing you where they are located within that depth. It doesn't tell you exactly that it's coming from a specific location, rather about a centimeter piece of root. But our hope is, like we would be able to use this and have an interface where when one uses the RhizoGrid indexing and have all of these omics layers, we can map them back to visualize and to pull out the interactions that you are interested studying along this rhizosphere system. And another approach we are taking is, this is one other example. So there's many more, when you have thousands of metabolite features, hundreds of microbial genera, so you can pick your favorite plant microbe interactions to visualize. So this is one other example where we found quinate in lower depths, quinate and microbe association. So we are visualizing the relative abundance of quinate and 4 different amplicon sequence variants along the roots, the same root system, to show that some are clustered to the crown, whereas there are others who are distributed along the root system. So there is a lot of dynamics that's happening in the rhizosphere. And I think, yesterday the keynote speaker said the root metabolome shapes the rhizosphere metabolome, and in turn kind of shapes the microbiome. So I think we are trying to get at that in a more spatially resolved way. So we've also generated data on, again, we were confident on the RhizoGrid derived molecular information. So we went ahead, we did a controlled drought experiment. This, again, this is still a work in progress, but some of the preliminary data, again, in this instance, I had 5 biological replicates for each condition. So we could have even more power as we try to piece apart the really biologically relevant information from all of the noise. So Winston, again, is taking all of this omics layers, and he's trying to use graphical neural nets, making all of these metabolite-microbe as nodes, trying to connect them to understand, is this enough information to extract drought-specific information, or is it enough information to extract depth-specific metabolite-microbe interactions? And he's finding some of these unique microbial metabolite pairs when he looks at the control well-watered versus water-limited drought conditions. So we are hopeful and we are excited about diving into some of this new data sets we've generated with the RhizoGrids. And I'm hoping, you will all, as you go through this user meeting, find ways that we can collaborate and do some of the rhizosphere work using some of these capabilities that we are building in EMSL. So with that, I want to thank everyone who was part of the work, and the funding and came from the Persistence Control SFA led by Rob Egbert. So I want to especially thank the SFA for allowing me to continue and develop and optimize the RhizoGrids. And I also want to thank them, where we build all of this capability and have used all of these analytical platforms to get at plant microbe interactions at a more spatially resolved manner. So with that, I want to thank you all for your attention and take any questions.